Hey everybody, welcome back to the class. In this video, we will go over Chapter 23, the Digestive System. And I will go ahead and warn you now, before we get started, this chapter is demanding. We will talk about a lot of hormones and a lot of enzymes in this chapter, and it's very easy for those terms to get confused with one another. So it's really important that you stay on top of this chapter, especially. And for those of you that like to make flashcards, take note of the very last two slides of the presentation. Those last two slides will have all of the hormones that we'll talk about, all of the enzymes that we'll talk about, all on one convenient table. All right, when most people think of digestion, they think of food in their stomach being chemically broken down and then sent on to the intestines and then out of the body, which of course is a part of it, but digestion also includes the mechanical breakdown of food. And what that means is taking a larger bit of food and turning it into smaller bits of food. So digestion is both the mechanical and chemical breakdown of food into a form that your cells can eventually use. So the process of digestion actually starts within the mouth as you are chewing your food. Now the digestive system will consist of a very long tube called the alimentary canal. And this is one long continuous tube that goes from the mouth down the esophagus into the stomach, small intestine, large intestine, and then out of the anus. That's all one continuous open tube. And in addition to that, the system includes all the accessory organs that will dump some type of fluid or secretion into it. So here is the whole system, both with the alimentary canal on the right hand side and then the accessory organs on the left. So again, you start with the mouth here, esophagus here, stomach, small intestine, then large intestine, and then out of the body. That entire length is all open to the outside world because you have the mouth on one end and then the anus on the other end. That's one continuous open space. And for the accessory organs, that includes salivary glands, liver, gallbladder, and pancreas. Now on this image, this shows the relative length of all the components of the alimentary canal, but the length from your tongue to the end of the stomach, that's about half of a meter. So that's about 1.5 feet in length. The length of the small intestine is about 6 meters. That's about 20 feet just in the small intestine. And then the length of the large intestine is about 1.5 meters, which is about 7 feet. So it's an incredible length of tissue folded over and coiled up all within your abdomen, especially between the small intestine and large intestine. All right, next thing we'll talk about is the alimentary canal in general. And it has the same basic structure throughout the entire length, going from the esophagus all the way down through the end point at the anus. So there are 4 basic layers that you'll find within the canal itself. You have the mucosa, submucosa, muscular layer, and the serosa. Yeah, you should be able to match these individual layers with what they do, and also on a image if it gets presented on a test. The first one is the mucosa. This will protect the layers that are below it by secreting mucus. And this is also the site of where things get absorbed within the alimentary canal. Just below that is the submucosa. Here's where you find vessels that will nourish the surrounding tissues. Next, you have the muscular layer. This is what will provide movement of materials through the canal. That's done by a process called peristalsis. And this is a reference to having a wave-like contraction. So moving a mass of material forward as a wave. And I'll show you an image of that in a moment. Then the outermost layer of this setup is the serosa. All right, on this image there are multiple things, but we'll zoom in on this area here. So this would be a part of the alimentary canal, one section of it. So we'll zoom in just on this one square so you can see all 4 layers. So the outermost layer here at the bottom, serosa. Then just above that will be the muscular layer. And then just above that you have the submucosa. You can see the blood vessels sticking out on that edge right there. And then just above that is the mucosa. That's where material will be absorbed throughout this canal, is in this layer here. And you can see these projections, these villi that are sticking up. And the feature that we talked about in the previous chapter, alacteal, those are the lymphatic capillaries that are found in the intestines. That's what you're looking at here in green. Then each of these projections will have micro projections that stick out on top of that, and they're called microvilli. So if this were to be presented on a test, you would most likely see an image like this one here. On this image, you're seeing an example of peristalsis in work. The term here, bolus, that is a reference to a a circular mass of material. And when you're talking about the digestive system, you're talking about food that's been swallowed as it's traveling from the esophagus and onto the stomach. So that's what this mass represents here. So for peristalsis, you have a wave-like contraction. So the muscular layer here is contracting, and that will push this bolus forward. So this image will lead to this, and then the area behind it will contract, and it will keep advancing the bolus forward until it reaches the entryway of the stomach. All right, like I mentioned a little bit ago, the process of digestion actually will begin in the mouth, and that's where you have the mechanical breaking up of the food that you're eating. And it's also where you will mix that food that you're eating with saliva. So that process, mechanically breaking up food and adding saliva to that mass of food, that process is called mastication. Also found in the mouth, you'll have the tongue, muscular organ that will help move the food down the pharynx into the esophagus. Remember, the tongue is what will force the epiglottis to close over so food that you're swallowing doesn't get into the trachea, into the lungs. Next organs we'll talk about are the salivary glands. Of course, these will secrete saliva, and that's there to help not only moisten the food that you're swallowing, but actually begins the breakdown of carbohydrates. And found within that saliva is an amount of mucus. And that's there to help lubricate the food so it goes down your esophagus much more easily. And also with mucus, you will find salivary amylase. And that salivary amylase is there to break down starches. So this is where you see the beginning breakdown of the food within your mouth chemically. There are 3 types of salivary glands. You should know what is unique about each one and then be able to identify them by image also. The parotid glands, these are the largest of the 3 kinds. These are found just anterior to the ears. You have the submandibular glands. These are found below your mandible, which is your jaw. So these are found on the floor of the mouth, just on the inside surface of the jaw. And then the smallest of these 3 kinds are the sublingual. These are found just below the tongue. All right, in this image, this is the person's tongue kind of pulled up at an angle. You have the parotid glands here, just in front or just anterior of the ears. The jaw is going to be right here. So right there would be the submandibular gland. And then just underneath the tongue here would be the sublingual gland. All right, next structure we'll talk about are the teeth. And the teeth are the hardest substance found in the body. And that's due to the enamel that covers them. Now, these are not considered a part of the skeletal system because they have a different structure compared to bones. That's why they're not covered when we talk about the skeletal system in Bio 168. Now, there will be 2 sets of teeth that you have in your life. The first set of teeth, the baby teeth, most people know them as. They're also known as the primary teeth or deciduous teeth. And for those, you will have 20. The adult teeth that come in after that are the secondary teeth or permanent teeth. And you will have 32 of those. And something I've mentioned in past classes, when you see terms like primary or secondary, remember, those don't indicate a level of importance. Those are indicating in what order those processes or those structures happened in. So primary teeth will come in first. Secondary come in after that. So your baby teeth are primary. Your adult teeth are secondary. Next structure we'll talk about is the pharynx and esophagus. Pharynx we talked about previous chapter with the respiratory system. As the cavity is posterior to the mouth, you'll find the trachea and the esophagus here. And neither part of the pharynx will digest food at all. The esophagus doesn't digest food. All it does is serve as a pathway, a passageway from the mouth to the stomach. And then in the esophagus, this is a straight collapsible tube that will help propel food from the pharynx down to the stomach. So people tend to confuse the details of the trachea and esophagus. The trachea is a rigid tube that's held open constantly with those C-shaped rings of cartilage. The esophagus will lie just behind that and is a collapsible tube. It only opens as a bolus is moving forward through it. If nothing is moving through it, it will stay collapsed. Now, the point where the esophagus meets the stomach has multiple names. Some people call it the lower esophageal sphincter. For this class, we will reference it as the cardiac sphincter. And this sphincter is what will prevent material from moving back into the esophagus once it's entered the stomach. So this image here, the esophagus would, of course, extend well beyond the border here. But right where the esophagus meets the stomach is the cardiac sphincter here. And remember what a sphincter is. It's a circular muscle that's able to close and seal off sections of an organ. Takes us to the stomach. A stomach is a pouch-like organ. It's about 30 centimeters long. It has multiple functions. I have 4 of the main ones here. Be able to recognize what is and what is not a function of the stomach. The first one, of course, receives food from the esophagus. Second, it will mix that food with gastric juice, which we'll talk more about in a second. It will initiate the digestion of proteins. And then once everything is done in the stomach, it will move that material onto the small intestine. There are some basic parts of the stomach you should be familiar with. The first one is the cardia. This is the small area near the esophageal opening. That's why I want to reference that as the cardiac sphincter, because it relates to the cardia part of the stomach. Then you have the fundus, which is a temporary storage area that's just above, just superior to the cardia. The main portion of the stomach is known as the body. Then you have the pylorus, which is it leads to the opening to the small intestine. So this is the last part of the stomach. Rugae, these are also known as the gastric folds. These are creases that you'll find on the inside lining of the stomach. And this is what allows for the stomach to expand with material. And then we had the cardiac sphincter at the top where the esophagus enters. There's also another sphincter that controls when material leaves the stomach. That's called the pyloric sphincter. So that will control how much and how often material will empty from the stomach into the first part of the small intestine. Right, so here we have the esophagus right here. Lower esophageal sphincter, or the cardiac sphincter, would be spanning that space there. Cardia, the area right here near that sphincter. Just above that would be the fundus, this area here. The main portion is the body. The pylorus is the last part of the stomach here. Pyloric sphincter, the muscle that controls what leaves the stomach and enters the first part of that small intestine. And then you can clearly see what looks like wrinkles in the inside of the stomach. Those aren't wrinkles, those are creases. So those are the rugae, or the gastric folds. So don't be surprised to see some image similar to this on a test in the future. All right, when we talked about the stomach, we need to understand how the secretions in the stomach work and what their names are. The innermost lining of the stomach has gastric glands, and each of those glands have 3 different types of secretory cells. You need to be able to match each individual cell to their secretion. The first one is probably the easiest of the 3. The mucus cells will produce mucus, and that's there to provide a very protective layer of the innermost lining of the stomach. The next kind of cell are the parietal cells. These are the cells that will produce hydrochloric acid, which is why the inside of the stomach is such a harsh acidic environment. In the stomach, you're going to have a pH of around 1 to 2, so very, very strong acid, and that is secreted by the parietal cells. The parietals also will secrete intrinsic factor, which allows for the small intestine to absorb vitamin B12. The third kind are the chief cells. This will secrete an inactive enzyme called pepsinogen. Now, pepsinogen only becomes active when it's mixed with hydrochloric acid. Then it gets converted to the most important enzyme found in the stomach, and that's called pepsin. That's also where we get the name for the soft drink Pepsi. If you weren't aware, Pepsi was developed by a doctor here in North Carolina, down at the beach at New Bern. He was developing a beverage to help cure upset stomachs. So what he developed, when he was looking for a name, he just took this enzyme, dropped off the letter N, and we get Pepsi. So be able to match what the chief cells secrete, what the parietals secrete, what the mucus cells secrete. So all of these secretions added together, so the mucus, the hydrochloric acid, intrinsic factor, pepsin, all of that combined is known collectively as the gastric juice. All right, for here, this image, you see all the 4 layers that we've talked about previously: mucosa, submucosa, the blood vessels, muscular layer, serosa. If you're going to zoom in on this area here, you'll see mucus cells, parietal cells, chief cells. On this image, this is really just showing how these 3 different types of secretory cells are arranged. Because these cells look so similar on an illustration like this, probably would not use this on a test. All right, now we will get into the regulation of gastric secretions. And it's at this point where material starts to get a little bit more demanding, because we start to include a lot more enzymes, a lot more hormones, and these can get confusing. But like always, if you do have questions on the material, please contact me and let me know. The first one we've already talked about in the previous chapter, somatostatin. And we talked about how it has a role with regulating insulin and glucagon in the pancreas. Well, it also has a role within the digestive system. It helps to inhibit acid secretion by the stomach. Sometimes you want that to be turned up, sometimes you want that to be turned down. It just depends on what is needed at that moment. So somatostatin will turn that secretion off. And the opposite of that would be gastrin. Gastrin will increase acid secretion. So whenever you don't need this gastric acid being produced, somatostatin will become more active and turn it down. Gastrin will turn it back up when it is needed. The next one is one that we will talk about multiple times in this chapter. That is cholecystokinin, also known as CCK for short. And this material is released by particular cells of the small intestine whenever proteins and fat enter the small intestine. It's there to decrease gastric motility. And what that means is, if you look at the size of the stomach, it can hold a good amount of material. You cannot have a full stomach emptying all that material into the small intestine all at once. You have to have a way to regulate how much enters the small intestine at any given time. A real world analogy, think of this as a doorman at a club. Certain people go in when certain people go out. You can't have everyone rushing the door all at once. So it's the same basic principle here. You're allowing a certain amount of material in at a time. And then when it gets too much, it stops that material from entering the small intestine. So that's one role that CCK has in this process. Next topic, gastric absorption. Even though the enzymes within the stomach start to break down proteins, the stomach is not well adapted at all to absorb anything. It's way too turbulent, way too violent, way too acidic of an environment to absorb anything. It's there to break down food and break down proteins and then move it forward into the small intestine. So of course the stomach is an important piece of this puzzle, but it doesn't really absorb anything because it can't. Then now we have the food you've already eaten, it's been through the stomach, it's been in the presence of gastric juices. At this point, as this material is leaving the stomach to get toward the small intestine, it's not called food anymore because it's not. It's now called chyme. So this is a semi-fluid kind of paste that's not the food that you ate, but it's not feces yet either. So it's not the finished product. It's that middle point. And then unfortunately, everyone knows what chyme is. If you've ever had an upset stomach, if you've ever thrown up due to intestinal distress, what you're throwing up is chyme. It's not the food that you ate previously, but it's not the finished product of digestion either. It's the midway point. So that's what you are throwing up, is a sampling of chyme. Food mixed with a collection of gastric juice. All right, next organ we'll talk about is the pancreas. Now I mentioned when the course first started, when we started with the endocrine system, I mentioned how the pancreas had a dual function. It is an endocrine and exocrine organ. Back then in January we talked about the endocrine function, and now we will go into the exocrine function of this organ. And the exocrine function of the pancreas is to secrete a digestive juice called pancreatic juice. Now the pancreatic juice is produced in very specialized clusters of cells called acini, and acini is plural. Acinus would be singular. So these clusters of cells, these acini or acini, release these secretions into tiny tubes, and those tiny tubes will merge with larger tubes. And then eventually you'll have one large tube that runs the entire length of the pancreas, and that will give rise to what's called the pancreatic duct. And it's this duct that will empty all of the pancreatic juice that's been collected into the small intestine. All right, here we have a internal image of the pancreas just behind the stomach. On here you can see the islets of Langerhans. We talked about those before. That will produce glucagon and somatostatin and insulin. These clusters here are just three small examples of the acini. Of course, there are going to be countless more, not just these three or four. These are found throughout the entire organ of the pancreas. In each of these clusters, you have a little bit of the pancreatic juice being made. Once that gets made, it gets delivered into this merging tube here. And this long tube that runs the entire length, basically collecting all the pancreatic juice that's been produced by all the acini. So all of that material that gets collected will end up here in the pancreatic duct. So all that material that's been collected will empty into the small intestine here. Now you'll find multiple secretions found in the pancreatic juice. They are there to help break down carbohydrates and fats and proteins and nucleic acids. You need to be able to match what each of these break down, and the names can get somewhat confusing and somewhat cumbersome. The first one, pancreatic amylase, will split glycogen into disaccharides. So we're talking about breaking up sugars here. Pancreatic lipase, that's the enzyme that will break down triglycerides. A triglyceride is an example of a lipid. On this slide, there is a lot of text, there's a lot of enzymes, but the one key item on this entire slide is this one, enterokinase. And I'll explain why in a second. So again, we're still talking about the individual components of pancreatic juice. Another one is trypsin. This is there to digest proteins. It is released in an inactive form, trypsinogen. It only becomes activated when it's in the presence of enterokinase, which is another enzyme. Chymotrypsin also will break down proteins. It's only activated when it's in the presence of trypsin. The last one, carboxypeptidase, also breaks down proteins. It's also released in an inactive form and is only activated by trypsin. So each of the bullet points that are listed here, trypsin, chymotrypsin, carboxypeptidase, all three have exactly the same function. All three will digest proteins. Each of them do it in a different way, but that's more detail than what we need. But all three of those have the same job. They digest proteins. None of them can be activated unless enterokinase is there to activate trypsinogen into trypsin. So if enterokinase isn't there, nothing else on this whole slide is even going to be activated. So you need enterokinase to turn trypsinogen into trypsin. That will activate chymotrypsin and carboxypeptidase. Another component of pancreatic juice is nucleases. These will break down nucleic acids. And the last component we'll talk about of pancreatic juice is bicarbonate ion. And pancreatic juice will have a very high concentration of bicarbonate ion, often referenced just as bicarb. And it's there to raise the pH of the chyme. And here's why. The chyme is coming out of the stomach. Well, the stomach is a very acidic environment. The pH of between 1 and 2. You can't have something that acidic entering another structure. It would just burn right through it. So you have to find a way to neutralize that very, very low pH. So that's what bicarbonate ion does. It is a buffer, essentially. It's going to add a very high pH solution to the chyme. So instead of the pH being 1 or 2, it can be much more tolerable to the small intestine. This is why ulcers within the stomach hurt so badly, because you have hydrochloric acid burning through tissue. That's why heartburn burns so badly, or small intestine ulcers hurt so badly, because you're having acid that can leave a chemical burn on tissues, physically burning through that tissue. So the whole point of bicarbonate ion is to raise the pH to neutralize the very low pH of the chyme. There are ways to regulate these pancreatic secretions, and there are hormones that we need to talk about. The first one is secretin. This will stimulate the pancreas to release the pancreatic juice that contains that high amount of bicarbonate ion. So secretin will regulate the secretion of pancreatic juice. One we've already talked about before, CCK, cholecystokinin. This is what will stimulate the gall bladder to release bile that it's stored. So here's what happens. You have the acidic chyme entering the duodenum, or the first part, small intestine here. The presence of that acidic chyme will trigger the release of secretin. So that secretin will stimulate the pancreas to release pancreatic juice, which one of its components is the bicarbonate ion, to help neutralize that acidic chyme. So the presence of the chyme is what will trigger the release of secretin, which in turn will cause the pancreas to release pancreatic juice to neutralize that chyme. Next structure we'll talk about is the liver. This is the largest internal organ that we have. The largest organ in general is your skin, but the largest internal organ is the liver. Of course, this will be just inferior to the diaphragm. Now within the liver you have a very unique circulatory feature. That's called the portal system. So all of the blood that is leaving your intestines through the venous system going back to the heart, it doesn't go to the heart directly. It goes to the liver first to be filtered one more time. And then from there it goes on back to the heart. So that's what the portal system is. It is another way to filter blood coming from the intestines. So here we have various images of the liver. Right lobe here, left lobe here. Is for reference the inferior vena cava up here. The gallbladder is just on the underside. If you were to look from the other perspective, you see it like this. And then we talk about the portal system or the hepatic portal system. Either term is fine. Here's what's going on. The small intestine will absorb whatever it needs. As the blood is going back to the heart, it will enter the hepatic portal vein into the liver. That liver will screen and monitor that blood that gets sent back. And then that blood will enter the general circulation again through the hepatic vein. So this is only regarding blood flow coming back from the intestines. It gets filtered one more time by the portal system. All right, when we talk about functions of the liver, it'd be difficult to name everything that it does. So we'll just talk about a few of them. It will produce glycogen from glucose. Now, this is the stored form of glucose. And it will break down that glycogen back into glucose when needed. Another role that it has, it will take part in the phagocytosis of old and worn out red blood cells or any other foreign material. It's there to remove toxins from the blood, like drugs, like alcohol, for example. So the liver is incredibly important and does a large number of tasks for the body. That's why it's such a major deal when the liver starts to fail. The next topic we'll talk about is bile. Bile is a greenish, yellowish kind of liquid that is made in the liver, but it is stored by the gallbladder. And within the bile, you'll find water, bile pigments, bile salts, electrolytes, and cholesterol. And if you have an excess amount of bile that's made, that's where you have the condition of jaundice being produced. This is where you have a yellowish coloring of the mucous membranes and the skin and the eyes. Think of a typical Simpsons character, that yellowish tint to them, but in real life. You can see that in their eyes, you can see that in their skin, and the darker yellow it is, the more serious it is. Having jaundice is a very good indicator that the liver is starting to fail. Now, the function of bile is to break down fat that's found in the food that you eat into very small droplets. Your body cells can't use fat in its normal form. It has to be broken up. It has to go through a process called emulsification. That's turning fat into very, very tiny droplets. And the bile salts that are found within the bile also enhance the absorption of fatty acids and cholesterol. Like I just mentioned, bile is stored within the gallbladder. Made by the liver, it's stored until it's needed in the gallbladder. And the gallbladder will only secrete bile into the small intestine when it is stimulated by CCK. So here's what's happening. You have chyme entering the first part of the small intestine here. When your intestinal cells detect the presence of fat, that will trigger the release of CCK here. CCK will stimulate the gallbladder to release bile, to break down that fat. So the presence of the fat will trigger the chemical that's going to break it down. The next organ we'll talk about is the small intestine. And this is actually where most of the work gets done in the digestive system. Most people think of only the stomach and nothing else. And the stomach, of course, has a role in this process. The heavy lifting, the real work of the system, gets done by the small intestine. And this is a long, very long tubular organ that extends from the pyloric sphincter at the end of the stomach to where it joins up to the large intestine. And this is what will complete the digestion of chyme, and also will absorb the material that your body needs. And then whatever it can absorb or doesn't need or doesn't want, the leftover portion will get sent on to the large intestine. And the small intestine has three components: the duodenum or duodenum, jejunum or jejunum, and ileum. And those are the order that they go in. The duodenum is first, and then the jejunum, and then the ileum. So we have the stomach up here, first part of the duodenum here, and then the jejunum here, and the last part would be the ileum. So all of this is around 20 feet approximately that's coiled over within your abdomen. Now there are countless tiny projections found within the small intestine, and they're called intestinal villi. It's there to increase the surface area. The more surface area it has, the more material it can absorb. And the term villi is plural. The singular would be villus. Each of these villi will have blood vessels inside it, and also nerve fibers and lacteals. Remember, those are the lymphatic capillaries. Also on each of these villi are even smaller projections called microvilli. So that's going to increase the surface area even more. So here we're looking at one villus with the lacteal inside and also the blood vessels. So for here we're looking at a real histological sample of a portion of the small intestine. This right here is one villus, just like it is here and here. And then on the edge of this you would see countless numbers of microvilli or even smaller projections. So this here where the cursor is the same thing as this. All right, you will find several enzymes that are found within the small intestine. You should be able to match what they do. The first one, peptidase. This is what will break down peptides into individual amino acids. The next group, they're all grouped together because they have the same job. Sucrase, maltase, lactase. All of those will break down disaccharides into monosaccharides. So that will break down sucrose and break down maltose and lactose. And then the last one here, lipase, that will break down fats into its individual components, the fatty acids and the glycerols. All right, next component is the large intestine, and it's called large simply because it is wider than the small intestine. It's not any longer, it's not even close to being the length of the small intestine. It's just a little bit wider. The first part of the large intestine is the cecum. This is where you find the appendix. Now, a term that you'll hear mentioned when it comes to the system is colon. Colon is referencing the large intestine only. The colon has different names for the different areas of the large intestine. And the name tells you in what direction the material is traveling. So after the cecum, the material will be moving up. So that's the ascending colon. Then it goes across the body. That's the transverse colon. Then it will turn and go downward. That's the descending colon. And then the last portion is a S-shaped area called the sigmoid colon. And then the rectum is the very last portion of the digestive tube. The anal canal, this is the last 3 to 4 centimeters of the large intestine. And then anus is the opening to the outside environment. So on this image we have just the large intestine. So here we have the ileum from the small intestine attaching here. So this first part of the large intestine is the cecum. You can see the appendix right here. The material that's in this area will start to go up. That's the ascending colon. Then it goes across. That's the transverse colon. Then it will turn here and go down. That's the descending colon. Then you have this S shape here. That's the sigmoid colon. And then you have the rectum, then anal canal, and then the anus. When people have a sigmoidoscope, the instrument will go into the sigmoid colon here. If you have a colonoscopy, it goes all the way up here, which is why you need to be sedated for that. Now the functions of the large intestine, it has very little digestive function. The biggest thing it does is it absorbs water and electrolytes. If it absorbs too much water, you're going to be constipated. If it absorbs too little, you're going to have diarrhea. It also will house the intestinal flora, the naturally occurring bacteria that we all have in our intestines. And that bacteria is absolutely necessary for many digestive processes to occur. And of course, the last function it has, it will form feces and carry out defecation. So at this point, whatever is being defecated out is material that the body does not want or does not need or cannot absorb. We will talk briefly about feces because you can't talk about the system and not mention it, at least in general. This is the material that is not digested or not absorbed by the body. Within it, you'll find mucus, water, electrolytes, bacteria. The coloring of feces can be altered by the presence or absence of certain bile pigments. So as unpleasant as it is to think about, you could learn a lot about someone's general health and their diet by their feces. Certain colors indicate certain problems. If it's a certain texture, if it's tarry or not. Do the feces float or not? If the feces are floating, that means the diet is really high in fat. All right, like I mentioned in the very beginning of this video, these last 2 slides here and here cover all of the hormones we talked about in this lecture, all of the enzymes we talked about in this lecture. It has their name, it has their source, and their function. So again, for those of you that like to make flashcards, this and this are going to be very, very helpful for you. All right, that brings us to the end of this chapter on the digestive system. Again, if you have questions, feel free to contact me either through email or by texting me. I do have those online office hours every Monday and every Wednesday morning, 9:30 to 11, or you can put them on the discussion board on Blackboard. Thank you for watching, and I will see you in our next video.